Good afternoon. I am Purya Tajra from KTH, and in this project, we are trying to understand the interaction network in the human immune system. This is a collaboration between Peter Brodin from KI and Dimos Dimorogounos from KTH. This project is motivated by three recent observations. First is that the immune cell composition, meaning the relative population of different cell types, varies a lot between individuals. Second is that this composition is highly stable throughout the person's lifetime as long as they are healthy. And lastly, this difference in the cell composition can actually predict differences in immune responses to certain stimuli. So how can we understand the dynamics behind this stability and predictable responses? The answer to this question is particularly relevant to immunomodulatory treatments, which basically allows us to predict if a person is susceptible to adverse effects from certain treatment. And ultimately, with good predictive powers, we can personalize treatments for each individual. So far, we have developed a network-based model where each node represents different cell types or other agents in an immune response, including the proteins and antigens. To identify the dynamics in this network, we need to answer two questions. The first one being, how are we going to represent the state in each of the nodes? We have so far seen that by looking at the cell populations alone, we can already identify some responses in the network. So we want to see that if we look deeper at the cells, like for example, the gene expressions, can we infer more about the regulation mechanisms between the cells? The second question is, how can we collect data efficiently so that we can infer the causality directions in the dynamics? In the Brodin lab, a long-term blood culture system has been developed that allows us to modulate the cells, basically meaning that we can remove targeted cell types and analyze the differences in the responses of the modulated samples. A first round of data collection is now done, and the data is being pre-processed, and we envision an iteration between modeling and data collection as we narrow down our hypothesis set. Two challenges have particularly stood out for us so far, and the first one is incorporating the existing knowledge base, what we know about the interactions between the cells that come in very different forms, such as differential equations, logical circuits, and agent-based models. And the second one is a lack of benchmarking for model evaluation, even though there is a growing body of research in multi-cell analysis. Looking forward to speak with you further on this.